So today we're covering Mendelian genetics, one of my favorite topics. I know you've heard that before, but I'll never forget the summer after I took genetics in college. I was so excited. I looked for some flowers I could plant in my parents' yard. I needed flowers that had only two colors. I needed flowers that the structure of the flower made very easy to reach the pollen structures. And I spent the summer, I planted red ones on one side of the yard, and the opposite side of the house, I planted white ones, collecting pollen, transferring pollen from one flower to another, collecting the seeds, very carefully putting them in an envelope and labeling pollen came from this kind of flower, and you put it in that type of flower. And the next summer I planted them, and you know what? It worked exactly the way Mendel said. It was a little bit over a year because I started sometime in June and I didn't get my flowers until late August or September. But boy, was that fun. And it was exactly what I expected down to the numbers. I love genetics, okay? We call it Mendelian genetics, everybody knows, because the father of genetics was Gregor Mendel. He was a monk and he was in an order that they were required to work. Some orders don't. And he was put in charge of the garden. Now, what is amazing, I have seen aerial pictures of the monastery, and it's actually a bunch of buildings with little courtyards between them. And they had, instead of one big garden, they had many small gardens in the courtyards, which was fantastic because it allowed them to isolate plants. They only pollinated the way he wanted them to pollinate because the pollen could not go over the buildings and come down the other side. They had plants that have been growing for hundreds of years, so it didn't take very long to end up with pure whatever he needed. It was just a case of being in the right place at the right time and also having someone with a tremendous brain, because he spent some 10, 15 years collecting data, and then he analyzed the data. Remember, this is 1800s. There was no computers. But he analyzed the data and he saw trends and came up with his famous laws of heredity. So it is really an amazing story, okay? We already talked about heredity as being passing characteristics from the parents to the offspring. So let's see what exactly did Mendel do. He worked with pea plants. Obviously, they had more plants in the garden, but pea plants were very convenient because he found 7 characteristics that he could study. You see in there that you have the shape of the seed, the color of the seed, the color of the flower, the shape of the pod, the color of the pod, how tall the plant was, and the position in which the flowers appear in the plant. But the interesting thing, and believe me, I had a hard time finding one characteristic with only 2 variations, and he found 7. Okay, but there was only 2 variations or 2 traits. So the seeds could be round or could be wrinkled. And if you let the seed dry, obviously it's going to wrinkle, but this was a fresh seed, still in the plant. Okay? The seeds could be yellow or green. The flowers were either purple or white. The pods, he called them inflated and constricted. Basically, the inflated means that you couldn't see the shape of the seeds inside. In the constricted, you can see it's kind of bumpy. You can see the outline of the seeds. The pods could be also yellow or green. The flowers could be axial, meaning they were all along the main stem of the plant, or they could appear only at the top, and they were called terminal. And then the plants could be tall or dwarf. So he found an ideal plant, and then he spent, like I said, years working on that. He began by wanting to have what he called true breeding plants, meaning that they always gave offspring with the same trait. Now, keep in mind these plants have been isolated in different courtyards, probably for a few hundred years. He still did several breedings where every time he crossbred a tall plant with a tall plant, 100% of the offspring was tall and 100% of the offspring of this one. So he still did it for several generations to make sure that it was really true breeding plants. And once he had true breeding plants, he began hybridizing, mixing them. So hybridizing means that he was crossing plants with different traits. The first generation that he began with, he called it the parental generation, represented with P. The first offspring, it was called the first filial. Filial means son, so that's the first generation that follows. So it's F1, and when he crossed this first generation, he got a second filial generation, F2. The example you have in here, you have some purple flowers and white flowers. When he crossed them, the next in the F1 generation, all of the flowers were purple. What happened to the white ones? Well, now he let them crossbreed the F1 generation, and in the second generation, he got purple and white, and they were in a 3-to-1 ratio, 3 purple for every white. And he did that over and over, and he did it with all 7 traits. Okay, here you have some of his numbers. We're not going to go through all of them, but you can see for all of them, it's a 3-to-1 ratio, 3 of whatever's in the left column to 1 in the right column. So obviously there was something going on in there. That's not a coincidence, agree? So he concluded that these traits, remember, these are the characteristics, they were controlled by some factors. He had no idea what it was. Remember, this is 1800s. But these factors were passed from one generation to the next, and every trait was controlled by 2 forms of the factor. And that allowed for all the different combinations. We're going to see examples, don't worry, okay? Of course, today we know that these factors are genes, parts of DNA that code for allele protein, and the variations within a gene, we call them alleles. So for instance, an example of a gene would be color of the flower. The alleles for the color would be purple and white. Okay? There's a few terms that we're going to be using all the time in this chapter, so you need to understand. Genotypes refers to all of the alleles, whether they can be seen or not seen. Just to give you an example, and it will be in the next page summarized, so don't worry if my handwriting is awful. But let's say that purple is going to be uppercase P, and I'll explain in a moment why uppercase, and lowercase p is going to be white. And we said that every trait is going to be guided by 2 factors, by 2 alleles. So you could have plants that are big P, some plants are big P, little p, some plants are little p. These are all the possible combinations. So the genotype is exactly what genes are present. Big P is a genotype, big P, little p is a different genotype, little p is still another genotype. Now, phenotype is what you observe. You don't see the genes, do you? But you can see, and the big P gives purple flowers. And be patient with me, we'll explain more about that. The big P, little p gives purple flowers too. The little p gives white flowers. So the phenotype is either purple or white. They're directly related. The genotype causes the phenotype, but they're 2 different things. And be very careful when you're doing problems. What are they asking me, the genotype or the phenotype? Because that happens all the time. I ask the students, write the possible phenotypes, and they give me colors. That's not, or give me genes. Give me the genotypes, they give me colors. Give me the phenotypes, they give me alleles. Read the questions, okay? Answer what they ask you. And I always tell students, don't answer something they don't ask you, because if you're right, there's no extra credit, because I didn't ask. If you're wrong, I cannot let it pass without taking some credit off. Okay? So now these alleles, some are dominant, meaning that they cover up the other one. Look, for instance, when you had the big P, little p, I said the flowers were purple. Why? Because the big P is dominant, that's why we represent it with an uppercase letter, and covers the little p that's white. So even though you have the allele for white, it's hidden. You don't see it in there. Big P is a dominant trait. Recessive trait is the one that's covered by the dominant. We represent it with the lowercase letters, and we are going to see that they appear. Well, actually, we saw one example. Let me go back in here. Remember, you have white, then the first generation, the white disappears, but then it reappears in the F2. You heard about traits that skip a generation? This is what we are talking about, okay? Children that look like the grandparent. There's something really odd about grandparent, and the grandchild gets it. Okay? That's what we're talking about. Now, whenever you have two identical alleles, that organism is called homozygous. Two identical alleles. So big P is homozygous, little p. But this is homozygous, that's upper P, so it's homozygous dominant. And little p is homozygous because they're identical, but it's recessive. Now heterozygous are when you have two different alleles, so big P, little p, that's heterozygous. So all of this is summarized in the next slide. I know my handwriting is awful, so I figured I'll just write down for you to read there slowly. So the first law that Mendel described was the law of dominance. He says, when you have a heterozygous organism, big P, little p, one trait conceals the other one. Again, when you have, in this F1 generation, remember you have big P from one parent, little p from another one. The purple hides the white because these flowers are all purple. Okay? The only way you can see a recessive allele is when you have two copies. That's what happens in here. If you continue, well, this is not the color of the flower, sorry. This is height. Can hardly see on the tablet. But this is height. So you have tall plants. I know they call it different. This is short or dwarf plants. So the first generation is heterozygous. Now you can have homozygous dominant, heterozygous, but it is only when you have two copies of the recessive gene that you can see a dwarf plant. The same thing that we did with the color, big P, little p, big P was purple. When do you see the white? When you have two copies of the little p. So you have to have two copies of the recessive allele for it to express in the phenotype. The first crossings we're going to be doing are monohybrid crossings, meaning that you're looking at only one characteristic. Okay? You begin with true breeding parents, always, but they're going to have different traits for one characteristic. What you get is called a monohybrid, and you predict the outcomes by listing all the possible gametes. So you are crossing big P with little p. Punnet square, you put the gametes of one parent. This parent can only make one type of gamete. I know I'm going to be repeating myself, but I think it's a good habit to get used to put all of the gametes, because if you start saying, I don't need to put this when the numbers are going to come out sometimes, and all of that, it's easy to just do it all the way from the beginning. The other parent can only make two gametes, so now you have big P, little p, big P, little p, big P, little p, big P, little p. You say, big deal, I could have done it on my head. Yeah, but we're going to come up with more than one trait, and maybe instead of the parental generation, we're going to cross the F1 generation. So it's going to start getting more complicated. Using Punnet squares assures you you're going to get the correct results with one condition. That's one thing you need to watch out. Make sure that your gametes are right. Like I say, in this case, it's very easy because each parent can only make one type of gamete, but we're going to get into slightly more complicated cases. If your gametes are right, then it's very easy to do the crossings. If the gametes are wrong, you're going to get it all wrong. So I always tell my students, when you're doing a Punnett square, put the gametes, stop, take a deep breath, check the gametes that they're right. If they're right, go ahead, you're going to do it right. It's not, you've done the difficult part. It's over. And then, of course, read the question. Are they asking you genotypes, phenotypes? Are they asking you for number, or are they asking you ratio? But as far as doing the Punnett square, the key is making sure your gametes are right, and everything works out fine. So you have an example in here, and I realize, at least I have a hard time reading this, but the parental generation, the yellow is big Y, and the green is little y. I prefer to write because I can make the big Y different from the little y. Okay, so this parent can only make one gamete, so this one can only make one gamete, so obviously the F1 is going to be big Y, little y. That means that if you get two of these offspring to breed, one parent is going to make the big Y, the little y, big Y, little y. So what are the combinations? Big Y, big Y, big Y, little y, big Y, little y, little y, little y. Notice that when you're listing the different genotypes, you have three different types of, three different types. You have the homozygous dominant, heterozygous, the homozygous recessive. But remember that Mendel said that the results he observed were 3-1? Yes, because everything that has the big Y is going to be yellow. You get 3 yellows for every green. But the Punnett square, again, if the gametes are right, will give you the results that you need, and all you have to do is, like I say, read carefully the question, make sure you answer everything they ask you. The next law was the law of segregation. This is almost common sense, but he talked about every trait being determined by 2 factors. You need to copy the factors. Of course, we know that we have 2 copies of each gene in a cell, and the 2 copies are segregated during meiosis. Remember, when we talk about anaphase 1, the homologous chromosomes separate. So that's why when you have 2 genes like big P, little p, or big Y, little y, they separate when they make the gametes, and you get big Y on one, little y on the other one. A test cross is a cross that you perform whenever you want to know if a particular organism is a homozygote dominant or heterozygote. Let's say a typical example that also explains the reason for doing that is deafness in dogs. Some breeds of dogs are more likely to be deaf. Everybody knows Dalmatians, they're very likely to be deaf. Okay, the problem with deafness is that it's a recessive gene, so you have a dog that can hear. Let's say that big D, I hate when it does this, I'm sorry. So when one day I'll get used to not, I'm sorry, just do that all the time. I write to cross to the edge. Okay, so let me just write down here. Let's say that big D is normal hearing, and little d is deaf. So you're a breeder, and you're thinking of buying this particular dog to add to your breeding group, and that dog has good hearing, but you don't know if it's homozygous dominant or heterozygous. If it's homozygous dominant, everything is fine, because you know the male that you have is homozygous dominant. But you don't want to buy heterozygous because some of the puppies will be deaf. Remember the 3-to-1 ratio. 1 out of every 4 is going to be deaf, and it's very difficult to sell a deaf puppy. right? Nobody wants a deaf dog. You can help it. So how do you find out? Well, what you do is you cross it with the homozygous recessive and see what happens. So let's cross this one with the homozygous recessive and this one. And it's very obvious that in this particular case, the offspring, they're all going to be heterozygous, so they all can hear. But in this one, you're going to have big D, little d, and little d, little d. 50% can't hear. So what do? You breed your dog. These are the expected results. And then you examine the puppies. If they all can hear, you know that dog is homozygous dominant, you want to keep it for breeding. But if half of your litter cannot hear, that dog is heterozygous, you don't want to use it anymore. You take the loss, you put the dog, hoping it's going to be okay, but it's not going to pay off. That's your business, so you just get rid of that dog. You sell to someone who's not planning on breeding and recover as much of your money as you can. So many times you want to test crossings because it's critical to know whether you have a homozygous dominant or heterozygous. Here you have another example with sheep. You have a black sheep, you don't know if he's homozygous dominant or heterozygous. So you cross it with a white sheep, that's homozygous recessive. And you say, okay, if, because we don't know, is homozygous dominant, then it can only make the big B genes. This one produces the little b, so all of your sheep are going to be black. Now, if he's heterozygous, it can make two different kind of gametes, big B, little b, that one's little b. So notice now that half of the sheep are black and half are white. So you actually do the crossing, observe the offspring, and then you can determine if this is homozygous dominant or heterozygous. Very simple. The third law that Mendel determined was the law of independent assortment, saying that genes, well, he referred to factors, do not influence each other regarding to the sorting of the alleles. This is something that's going to become very important with dihybrid crosses when you have two characteristics to make sure that every characteristic is independent from each other. And the best way to explain this is going to be to do an example. Okay? But this is usually the point where most students make mistakes. So listen very, very carefully. So we have a dihybrid cross, and I got a picture that's big enough. So you have two heterozygous, big R, little r, big Y. So it's round and yellow, and another big R. What I'm saying is that let me put it like what you have in the gametes only one allele. Okay, I have a students write one gamete to be big R, little r. No, because you should have only one R, or little y. No, you have only one. You're going to have big R, or you're going to have little r. Only one. And you need to have one of each trait. Because if you tell me one gamete is big R, I say, fine, but what color? Is your P transparent? Or some people put little y. Okay, that's fine. We know it's green, but is it round or is it wrinkled? I don't know. You need to put only one allele and one of each. So you go there and you say, okay, what are the gametes? The big R can combine with each one of the two Ys. So you have big R, big Y, big R, little y. Now you go to the little r, and again combines with little one, little r, big Y, little r, little y. And you do the same thing for the other parent, and now it's just a matter of crossing. And by the way, this is another number that I don't know if anybody will actually ask you for the number, but if you look at the round yellows versus the wrinkled yellows versus the round green, Sprinkle green knows the ratios 9 to 3 to 3 to 1. Like I say, I don't know if anybody's going to actually ask you what is the phenotypic ratio, but when you're doing a problem, remembering that number allows you to really quickly count and give you an idea if you're right or wrong. But once again, if you got the gametes right, you're going to get it right. It's very, very difficult to make mistakes when you're just combining, say, big R, big R, big Y, big Y. The key is putting the gametes right. And like I said, the most common problems would be to put only an R or a Y. No, you have to have one of each. Or to put two R's or two Y's. No, one of each, but only one. If you get that right, the rest is easy peasy. I promise. And that's what Mendel was saying. The color and the shape are independent from each other. So I have a worksheet with a bunch of genetics problems. If you like to practice one-on-one, if you like to do my worksheet or something like that, go to the video description, click on the link to my WhatsApp profile, and make an appointment. I will be glad to share with you my problems and work with you.